I'll get started. Okay, so thanks for coming. So today I'm going to be talking about developmental evolution using this model, which is the marine annelid Strebospio benedicti. And so my lab works on Strebospio as a model because it actually has two different types of development contained within a single species. So there's two different types of females, and in each segment they make eggs. And what you can see is that on the top female, that is making hundreds of small eggs, and the bottom female is making dozens of large yolky eggs. And there's some differences in how these eggs develop, which I'll talk about in just a minute. And there's some really big differences in terms of the larval types. So these small eggs, they don't have enough maternal investment in order to make an offspring that can metamorphose and turn into a juvenile without exogenous feeding. So these top row, these planktotrophs, are obligately feeding larvae. And they spend about 3 weeks out in the water column, swimming around, eating, before they can metamorphose into juveniles. And on the other hand, the bottom female with the large yolky eggs is making less lithotrophic or yolk-feeding larvae. These larvae, once they're released from their mom, are only larvae for about a day, and then they are competent to metamorphose into juveniles without feeding. So there's all sorts of differences between these larval types in terms of egg size and fecundity, also larval development, but also just larval ecology and dispersal ability as well. But despite all of these life history trade-offs, by the time they metamorphose into juveniles, they're morphologically indistinguishable. And as adults, they occupy the same niches. So what we have here is a dimorphism that's really only contained in the developmental stages. And what's really important to remember about Strebospio is that these individuals cannot switch back and forth between which type of offspring they're going to make. It is set, it's fixed, it's highly heritable, so this is due to genetic components. And so what that means is we have a really rare opportunity here to look at gene expression differences and how they contribute to development within a single species. So instead of comparing development across lots of different species and trying to understand how gene expression has evolved, we're looking at basically the most microevolutionary scale, right, within a single organism or species. All right, so before I talk about gene expression differences, I just want to emphasize some of the differences between these larval types. So on your left, that is the less-lithotrophic morph, and on the right is the planktotrophic morph. I'll refer to them as LL and PP in orange and green respectively. So you can see there's a really big difference in terms of size. So the less-lithotroph is much bigger than the planktotroph, which makes sense because there's an 8-fold difference in egg size there. But it's not just a size difference, right? The planktotrophs also have some key morphological differences as well. And the one that I'm highlighting here is Chaetae from the Chaetal sacs. So these are long swimming spines that they have, and they have a startle response. So they flare them out to deter predators. It helps with buoyancy. It helps with particle capture. And so we think that these Chaetae are probably under strong selection and necessary because, again, this is the morph that's out in the water column for 3 weeks, subject to a lot of mortality and predation. And the less-lithotroph never makes these Chaetae at any point in their development, right? So it's not just like a scaling or size difference. There's some key morphological differences as well. All right, so we are interested in understanding how gene expression changes between these two types during development. And to do that, we first had to establish the embryogenesis and larval developmental timeline for Strebospio. So I'm showing that here. And Strebospio is a spiral cleaver. So like most annelids, it goes through these very stereotypical stages. And the only point where you can start to really see divergence between the planktotroph and less-lithotroph is much later towards the larval stages. But they reach the same embryological stages at almost the same time and at different sizes. So there is a difference in time, and that is because the less-lithotrophic egg is much larger, so it takes longer to divide. And so at the end of this, the less-lithotroph actually lags behind the planktotroph by a few days to reach the equivalent stage. So what we wanted to do is look at how gene expression changes during development. So we picked 6 equivalent time points. So we chose the 16-cell stage, blastula, gastrula, this prototroch stage, swimming larvae, where you can again see these morphological differences, and then 1-week-old larvae, so 6 time points. And I should mention this is all entirely work done by a grad student in my lab, Nathan Harry, so this is one of his chapters of his dissertation. So using these time points, we wanted to ask, what are the gene expression changes that underlie developmental differences? So for each of those time points, we sequenced 5 or 6 replicas of each type at each time and did bulk RNA-seq. So we see that there are over 14,000 genes that are expressed during development, and we're just plotting them here in PCA space. You can see PC1, which explains most of the variation, is developmental time, going from 16 cell up to 1 week. And then the samples cluster together fairly nicely, which is always encouraging. It's lovely when samples cooperate. So what we see is that out of those 14,000 genes, how many of them are differentially expressed between the planktotrophic and less trophic morphs? We break that down, and we see that only 36%, so about a third of the genes that are expressed are actually differentially expressed between the two types. And most of that is pretty early in development. So about 25% of the genes are differentially expressed at the 16-cell stage, but the magnitude of that difference is not very large. Right? So lots of things that are differentially expressed a little bit. But that drops dramatically as we get to the gastrula stage. So at this point, only about 4% of the genes are differentially expressed, but that magnitude is much bigger, and that tends to stay throughout the rest of the larval development. So potentially, these are genes that are contributing to those larval developmental differences. All right, so with this data set, one of the things we wanted to do is really kind of dig in and classify what differential expression means and how we can measure that. So what we did is we made 3 categories for differential expression. The first of which is called heteromorphies, which is the change in the amount of gene expression. So this is where you have a gene that's expressed both in the planktotrophs and lecithotrophs, but has roughly the same shape of expression. So for example, here you can see that both the planktotroph and lecithotroph have a peak around gastrulation, but the planktotroph has significantly more expression than the lecithotroph. So that's a change in the amount of expression, but not necessarily the timing. The next category we're calling heterochronies of gene expression. So this is a change in the time of expression, and that would be where the peak of expression changes between the planktotrophic and lecithotrophic morph. We have a bunch of statistical analyses we go into as to how to quantify those heterochronies and how big those heterochronies are, but I don't have time to get into that today. And then we have a third category that we're calling morph-specific expression. So these are genes that are only ever expressed in one of the 2 morphs at any point in development. So only expressed in planktotrophs are only expressed in lecithotrophs. Now, as developmental biologists or evolutionary biologists, you might have some a priori expectations about how often these categories occur in our comparing between our larval types. I'm just going to jump to the punchline here and tell you. So what we see is that like 46% of the differentially expressed genes are heteromorphies, and that probably makes sense. That's like the least dramatic difference in gene expression. We also see 29% are heterochronies, so they're changing in the timing of expression, either getting turned on earlier in planktotrophs or earlier in lecithotrophs. And then we do see that 25% of the genes are morph-specific, so they're only ever expressed in one of the 2 types. I think that's kind of high. That's a lot. Again, this is a single species. They have the same genome. They have the same genes. It's just only ever turned on in one of the 2 types. But of course, one of the reasons that we did this study is we wanted to categorize this, right? There's not a lot of other studies that we can reference where we've got interspecific comparisons of development. So we essentially wanted to do this to sort of lay the baseline of like, what do we expect for differential expression, again, when you're looking at this really microevolutionary scale. So at least now we have like a reference point for how much differential expression to expect. But of course, we're not just interested in doing all these statistical analyses and trying to quantify expression differences. We're really interested in what genes are affecting these developmental differences and how are they regulated. But as anyone who's ever done RNA-seq experiments know, going from a list of differentially expressed genes to a list of candidate genes is kind of an art form. And so what should we pick, right? Should we look at the more specific genes? Are those the ones that are definitely contributing to developmental changes? Should we look at ones that have really big differences in heterochrony? What we know is that sometimes really big differences in gene expression might not translate into phenotypic differences, right? So it's kind of a conundrum to think about what genes to chase from this sort of analysis. But I think Nathan was quite smart about it, and he decided to do WGCNA analysis, which is weighted gene co-regulatory network analysis. And again, I don't have time to go into this, but what it does is it groups our genes into modules based on co-expression. And what he found is that there are 68 modules in our data set of co-expressed genes, and we can associate these modules with a trait of interest. And in our case, the trait that we care about is planktotrophy or lecithotrophy, right? And so we find that there are 2 modules that are significantly associated with larval type, and one is in the planktotrophic direction and the other is in the lecithotrophic direction, which is, of course, very convenient. When we dig into those modules, we find that there's a really manageable number of candidate genes here. So 65 in the planktotrophs and 21 in the leucotrophs. I've thrown the Go terms up here basically just like for our amusement. Go terms, especially in the Spiralia, can be somewhat useful. For example, the top one under leucotrophs is bioluminescence, but they don't do that. So we have to dig in and figure out what these genes are actually doing in our system. But some of them are incredibly helpful and we already know are important. So one of the ones that I've highlighted here is chitin biosynthetic processes. And we know that this is an important set of genes because, again, one of the key morphological differences between the types are these swimming spines, the chetae, and this is made of chitin. And we know in the annelids that the gene responsible for that is chitin synthase, right? So we expect to see differences in expression in the chitin synthase genes. And in particular, when we pull out one of those genes, chitin synthase 2, we see exactly the expression pattern that we expect. It's basically not expressed in leucotrophs during development, but it really peaks in the planktotrophs right as they're making those swimming chetae. And it drops off when they become juveniles and they're no longer making those swimming chetae. So we basically see the exact pattern that we expect in expression. So one of the things that Nathan was able to do was then to generate probes for chitin synthase 2 and other chitin genes. And he's using HCR, so hybridization chain reaction in situs, and looking to see exactly where and when the expression differences are happening in the larvae. So here you're looking at the planktotrophs at the very early stage, and I've highlighted the ketal sacs in yellow, and hopefully you can see that there's some bright red chitin synthase being expressed there, right actually in the ketoblasts that are making the chetae, which is very exciting. That stays on through later larval stages and then disappears during the juveniles, which I don't have here. But then if you look in the leucotrophs in the same ketal sac location, you don't see expression of that gene. So we do see that this is a gene that's like a key developmental difference between the larval types. So chitin synthase 2 is the first gene we've identified that actually changes the larval morphology between the morphs. And we're very interested in trying to figure out exactly how that works. One of the things we're working on right now is functional validation, like knocking it out. We're making some progress there. But I think the really important question is, what are the upstream regulators, right? So again, these two larval types have the same genome. They both have chitin synthase 2, but what is turning it on in one morph and not in the other, right? So this is the exploration that we're really interested in going forward. And we have a number of different approaches that you'll have to stay tuned for because we don't know yet. But what I want to say just in conclusion is that looking at this differential expression analysis, we see that about a third of the genes are differentially expressed during development of these two types, even within a single species. And of those, only about a quarter are expressed in just one morph. There's very few genes that have major differences in their expression patterns between the two morphs. And the RNA-seq analysis really does allow us to get to some unbiased candidate genes that are key players in developmental differentiation in the single species. All right, and with that, I would just like to thank the people in my lab. As I mentioned, this is all work done by Nathan Harry, a PhD student. And I'd like to thank everyone else who's worked on this and my funding. Today, I just talked about a development project, but we also have population genetics that we're looking at, comparative genomics, many other projects in our lab. So I am recruiting. If this is interesting to you, please just reach out and let me know. All right, thank you.
All right, and it's 11:30, so thanks everyone for coming to